Okay, good afternoon, everyone. First, I want to thank the organizer for giving me the opportunity to report our result. Actually, for this data set is a member of MUQC and the SEQC consortium needed by FDA. I have been dealing with data set nearly 10 years. This is just one of the results when I work on this. Okay, I think for this Neuroblastoma challenge, the major goal is one is for the effective data integration. The second is no matter what kind of method you want to use, the ultimate goal is to try to increase or improve the survival time prediction. As a previous report have already point out for the neuroblastoma, I think the remarkable heterogenesis make this prediction really difficult. So for us, our pipeline and the data summary, we first use the gene expression data only to build the model. Next, we use the copy number variation data only for the second model. Last, we combine both of the data and build the third model. That's our process. For the gene expression data, they totally have 498 data set. We only get the 496 data. So we select half of them randomly from and get the 248 data set as training data. Leave the rest of them for the testing. So we run this Cox regulation model for each of these 46,000 genes and select genes with FDR less than 0.1 for next analysis, which leave us to get around 8,814 genes. These genes or the potential will be signature for this model. Next, we use the middle value for each of these genes to divide the patients into two groups and test the survival between these two groups. And what lead us to select the genes with FDR less than 0.1 for subsequent analysis. Eventually, we get 6,382 genes. Each of these genes could be potentially a single biomarker for this modeling. So last, we run this correlation between this candidate gene and the survival time, and we eventually get the 48 genes with FDR less than 0.5, and after the Cox regulation modeling and stepwise regulation, we eventually get 5 genes, which is we use this 5 genes to build the risk score model. Based on this model, we can just predict this training set. Among them, there's 1/3 of this patient can be defined as high risk, which is consistent with previous clinical data set. For this test data, we apply this risk score model. For this test data set, we also get that 1/3 of the patient is high risk. As the accuracy is around 83% for the test set is really consistent with this training set. So next, based on this cost model, we plot this Kaplan-Meier curve with a log-reg test. You can see for this, based on the survival, we can separate these patients very well based on the training set and the test set. So for this endpoint is the risk status. Compared with the clinically defined risk status, our modeling can get the precision is around 81% for training data set and 78% for test set. The record is around 84% for training set and 83% for test set. For those patients matched with copy number variation data, that's around 138 samples. The precision is around 46% for training set and 76% for test set, and record is 0.84 for training set and 0.72 for test set. You can see for this CNV data set, since the prediction power is less than the whole data set. So interestingly, for this cost modeling, our model can also predict this, divide this high risk group into two subsets. It's the same as this no risk. I think previous Tina has also showed us they selected the gene set can also separate this high risk into two subsets. It seems this result is consistent with each other. Yeah. So next, we work on the copy number variation only model. Use the same process. We selected 3 copy number variation no side. Those interesting is that those 3 CNV no side is not located in any genes. That's in the genic region. Based on this model, we built this Cox model, and it seems for the precision is less than 0.5 training side and test side. For the record, it's roughly around 50% for the training side and the test side. That means for this CNV, the copy number variation data set, the information is not so good to build this model. So next, we integrated this features selected from expression data and the CNV data, and build the final cost model with those 8 features. For this integrative, this Cox model, the precision is around 45% for training side and 68% for the test side. The recall is around 48% for training side and 90% for test side. So next, we also use the logistic regression and the Lasso model for this endpoint favorite status. We use the 5-class validation process. You can see for the RNA-seq based model, the average precision is around 95%. And the average recall is around 85%. For CNV only models, the average precision is around 85%, and the average recall is around 65%. For the combined model, the average precision is around 95.8%, and the average recall is around 90%. It's a margin increase a little bit for this integrative modeling. So in summary, we built 3 models for cost by Cox proportion, hazard regression model, and used overall survival as endpoint. We also built 3 models by logistic model, regression with Lasso regularization to predict patient favorite status. The result is show the clinically defined risk status is not very good classified for prognosis. But the combined classification result for RNA-seq data and this clinically defined risk state, we can reach this, the risk state can reach this AOC is around 80% for the combined model, and the gene expression model is obviously outperforming the CNV only models in both studies. Therefore, we believe as the more CNV data needed, so that could potentially improve this prediction power. So because we have dealt with data set nearly 10 years, I just a discussion with the several friends in this consortium a moment ago. Since we got this data set for the whole consortium, I think over several hundred models has been built to predict this endpoint. This accuracy is not so good. That makes me think about probably the problems come from this clinical data set, not our modeling. Because so many people have been involved with this project and built so many models, the accuracy is not so good. I think recently there was a paper published, it's indicated for pathologists. Normally, they identify these patients, the accuracy around 80%. That makes me think about probably we can just connect this consistent misclassified samples and get this back to the pathologist. Let them recheck this patient sample. Probably they can correctly this misclassify based on these patients. So last, I would thank my students for this project.
Thank you, Tieliu.